Hi guys, this slide deck covers chapters 13 and 14, parts of chapters 13 and 14, and focuses on RNA, especially transcription and processing. So taking the information from DNA, transcribing it, reading out that information, and some amount of processing of those RNA molecules. And in this figure, this figure is interesting. It shows DNA here. I'm kind of tracing it with a pen tool here. And then it shows all along that DNA RNA molecules that are being synthesized coming out from the side. So in each position here, the DNA is being read by RNA polymerase and a new mRNA is being produced. And that mRNA might be used to regulate other processes. It might be used read by a ribosome to make a protein, whatever the function of that RNA is. But this is showing the transcription in action from those DNA molecules. So something you may have heard before is the central dogma, and this is the idea that DNA codes for mRNA, which codes for protein. And the key processes here are transcription and translation. And transcription you can think of as in a courtroom, you have a transcript being produced by a stenographer. You have um, oral arguments, for example, in a court case being written down and in notes. And so you're taking one language, you're taking English, but you're going from the oral form to the written form. So you're staying in the same language of English in that case, versus what happens during translation, going from mRNA to protein. In translation, you take one language and turn it into another language. So, from English to Spanish or from Arabic to Farsi or whatever it is, you're going from one language to another. And mRNA, you are taking the language of nucleic acids and translating it to the language of proteins, which is amino acid sequences. So that's the central dogma of molecular biology. We'll focus here on transcription, and later presentations will focus on translation. So transcription can result in all sorts of different types of RNA. We're going to focus mostly on messenger RNA, but just to be complete, I wanted to show this table, and this comes from the textbook in chapter 13, on all of the different types, or many of the different types of RNA that can be produced. So ribosomal RNA is a really important type of RNA and the most abundant in cells. It's the most prevalent type of RNA that's in cells. It's the RNA that's in the ribosomes. And then there's message RNA. That's the RNA that's going to code for genes and be ultimately translated to proteins. Transfer RNA brings the amino acids to a growing polypeptide chain in the ribosome. Talk more about that later. And then down here, there's a whole bunch of different regulatory types of RNA. We won't have time in this class to go into the functions of each of those, but nevertheless, they're very important. And the end of the chapter goes into a little bit of coverage of some of those different ones. But we're really going to focus in this presentation on the role of messenger RNA in particular, because that's how in many ways the information that's encoded in DNA. That's how it's eventually used and turned into protein. And these are just some questions you can take a look at. Basically, just says after reviewing this slide deck in the textbook, you should be able to answer some questions about transcription, and these questions are below. So if you want to use this a review tool, you can come back here and take a look at this slide and see if you can answer these kind of in your own words. What's the purpose of translation? What are key molecules that are involved? What do the terms template and non-template strand mean? What determines the starting point of transcription? That's a very important one. How does a cell know where to begin, where to set down the transcription machinery on the DNA molecule? And then last, what are the stages of transcription? What happens at each stage? So we'll be covering this information here in the slide deck. RNA polymerase is sort of the workhorse enzyme of transcription. And you've seen other similar types of molecules in previous presentations. For example, when we've talked about DNA replication, there's a molecule called helicase. It unwinds the DNA. And there's DNA polymerase. It synthesizes new nucleic acids. RNA polymerase does both of those things and more. And so you can think of it as sort of a multitasking enzyme. It opens and unwinds DNA so that it can move along the DNA molecule. It copies mRNA from a DNA template. The DNA closes behind it. It doesn't have to have any sort of pre-existing nucleic acid. So DNA polymerase requires an existing nucleic acid, so you can hook the new nucleotide onto the hydroxyl group, the 3' hydroxyl group. RNA polymerase doesn't need that. But one thing that is similar is that the synthesis of mRNA happens in the 5' to 3' direction, similar to DNA polymerases. So synthesis happens 5' to 3'. But RNA polymerase is the key enzyme involved in making RNA. It's adding new RNA monomers, right, ribonucleotides, to a growing RNA template, growing RNA strand. One thing that can be confusing, talking about transcription, is what's the template and non-template strand. And I think this figure shows it well. Sometimes I think of some synonyms for these words as a little bit more straightforward in the coding or non-coding strand. When you look up a gene, let's say you go online and you want to know the DNA sequence for some gene, you are presented with just a single line of A's, T's, C's, and G's from left to right. What that represents would be in this figure below, the non-template strand on the very top, or sometimes called the coding strand or the sense strand. So for example, if I wanted to know my my DNA sequence for some enzyme like alcohol dehydrogenase or something like that, or TRPV1 or TRPM8 or any gene that I was interested in, I would look up that sequence and I'd be presented with a sequence of DNA left to right, like it's on top, CTG, CCA, TT. The way that relates to what's actually produced is exactly equal to the mRNA that would be produced. So down here in red, except you switch out the T's for U's because RNA has uracil instead of thymine. So again, the non-template strand is the coding strand or the sense strand. The other one here, the template strand, that's what's used to make the coding strand. So it's sometimes called the non-coding template strand or the antisense strand. But the key thing to remember here is the coding strand or the non-template strand is exactly equal to the messenger RNA molecule that you're interested in making, except switching those uracils for thymine. Okay, so we'll go through the stages of transcription. And these stages are initiation, elongation, and termination. And before we get into that, we'll look at the structure of a gene first and begin with initiation. So the structure of a gene you could diagram out like this. You have from left to right, and sometimes we say that this region would be upstream of the genes that we're talking about, and this region would be downstream. From left to right, we have a promoter and an operator, which are indicated there. And these are regulatory regions of the DNA molecule. So this is showing a DNA molecule with a promoter and an operator. Talk about what those are in just a second. They're regulatory regions. And then it shows a block of genes, gene A, B, C, D, and E. So in bacteria, oftentimes they'll have their genes packed tightly together, and so that if you kind of initiate transcription of one of them, a bunch of them all get made that are related. So this is showing something called an operon. So this only exists in prokaryotes, and it's specifically called a lac operon. And this gets switched on when there's lactose to use. So there's different types of sugars. There's glucose, there's lactose. When lactose is present, bacteria will switch on the lac operon, and that turns on genes like lactase, which can break down lactose. It also switches on genes to code for lactose transporters to help bring in lactose into a cell. So it's a whole bunch of functionally related genes that all get switched on at the same time. So to be clear, operons are sets of functionally related genes. Upstream of that is the promoter, or towards the 5' side of the gene. And the promoter is where RNA polymerase binds to initiate transcription. RNA polymerase is going to begin to unwind the DNA at this region. So the promoter is actually a section of DNA that other molecules can recognize and bind to. And it's not coded for, so the promoter doesn't end up coding for the protein in question or any of these proteins that are encoded by gene A, gene B, gene C. It's just a region of the DNA that's upstream of those genes that the RNA polymerase can bind to. And then the operator is one of several different types of regulatory regions that can exist that kind of acts like an on and off switch. So other molecules can bind to the operator and help potentially affect the amount of mRNA that can be produced from this. It can, in some ways, you can think of it like if you just stick a molecule there that just blocks the promoter from binding, or enhances, in some cases, promoter binding. That can be the function of DNA sequences like an operator. And then on the 3' side, or the downstream side, is the terminator sequence. And this is a special sequence of nucleotides that helps the RNA polymerase release from the DNA template. So this is the structure of how many genes in prokaryotes are organized. And in eukaryotes, they're very, very similar. The main difference would be that in eukaryotes, there would just be a single gene present instead of a set of functionally related genes. And that gene would contain, in eukaryotes, introns and exons, which we haven't defined yet, but we'll talk about that later. This is a link to a nice video that shows transcription in an animation that I think is helpful. So if you want to take a second to watch that, I think it's helpful to have a visual of this process before we keep going. Okay, so zooming in a bit on prokaryotic gene structure. So again, this is a different graphic, but the same idea, that there's some sort of coding sequence of genes labeled in blue here. And upstream of that, or 5' of that, is the promoter. So let's look a little bit more in detail at the promoter sequence. We'll talk about some of these other things that are involved. So here, the promoter sequence, which is just sort of approximated by this yellow bar along this DNA molecule, is then expanded to indicate the actual sequence of DNA that's present in the promoter region. And there's a few things to notice in this. There is the transcriptional start site, +1. It's called the +1 sequence. That's where the actual transcription starts. That's where you start building your mRNA molecule. And the circle here kind of obscures it up here to the right and below the promoter region, but that's that plus region here. The transcription start site is there. And just to the 5' side or upstream of that are what's known as the consensus sequence. And this can differ species to species. But what that means is that if you were to sequence the DNA for many, many different types of genes, these are all genes in E. coli. So these are shown over here on the left side. RRND1, RRNX1, RRNE1. So these are all just different genes. And you lined them up, you would find some commonalities. You would find that approximately 10 base pairs upstream of the transcription start site, and you'd know this because your mRNA molecule would begin here. So you could sequence your mRNA and it would start here, but the sequence of the DNA would contain, just to the left of it, in almost all cases, a T at this location, and then followed by an A and a T, and then sometimes a G or an A, and then another A, and then a T. So in almost all cases, E. coli genes, 10 base pairs upstream of the transcriptional start site contain this sequence, TAT-AAT. And if you look even farther upstream, 35 base pairs upstream, in fact, of the transcriptional start site, you find this consensus sequence again. It's the consensus of many, many different genes, if you want to think about it that way. It's in agreement. You'd find that consensus sequence is TTG ACAT. So a consensus sequence is derived from not really an average, but just looking to see, hey, what's typically at that base pair position at that location, upstream of a lot of genes. So upstream of the transcriptional start site, 10 base pairs and 35 base pairs. You see these different consensus sequences. So that's what the promoter actually is. It's these specified sequences of DNA. And this up here at the top of this image, it shows that transcriptional start site with the +1, and then the messenger RNA begins, the sequence of the messenger RNA begins, RNA polymerase starts churning out a molecule, but some of that isn't translated. So this 5' UTR stands for the 5' untranslated region of the mRNA molecule. So that's where the messenger RNA it exists, the RNA molecule itself exists, but not all of that mRNA is translated. You don't see translation until you hit a start codon. We'll talk more about codons in later presentations, but one sequence that can indicate translational start is ATG. So codons are 3 nucleotide sequences that are recognized at ribosomes, and ATG codes for a methionine amino acid. It's often the first amino acid of a gene. And so then as you go to the right, you would just continue producing your mRNA transcript. So now just some labeled slides in case it's convenient. Transcription starts at the transcriptional start site. Blue here indicates the first transcribed base. But translation of a protein doesn't start until ATG, until the translational start site. This gap here that's indicated in pink, this is the 5' untranslated region of a gene. And again, the message RNA contains that sequence with U's instead of T's, but that doesn't end up being translated at the ribosome. You don't start translation until that start codon is encountered. Okay, So we won't spend a lot of time on this idea, but just to be complete, it's interesting because genes can be encoded in either direction in the genome and on either strand. And so it can get a little confusing because the coding strand is defined by the mRNA sequence that it produces, but one strand could be the coding strand for one gene, but the template strand for another gene. So they're not always the same if they're for those different genes. So for example, gene 1 is encoded on one one molecule, one strand of that double helix DNA molecule, but genes 2 and 3 are encoded, in fact, in the opposite direction on the other strand of that DNA molecule. All right, so we'll talk a little bit more about how you start transcription, initiate transcription. We talked about the structure of a gene, now we'll talk about how you actually get going on it. And uh, in prokaryotes, RNA polymerase, and this is true in eukaryotes too, is composed of a number of separate, smaller protein subunits. Prokaryotes, it's a simpler structure with fewer subunits, and in eukaryotes, it's many more subunits that are present. But in prokaryotes, the core of the core of the RNA polymerase enzyme is a couple alpha subunits, an omega subunit, beta subunits, and these have different functions. The alpha and omega, these interact with other proteins. The beta is actually doing the transcription that's happening. The beta prime subunit, it's binding the DNA and holding the molecule along the DNA molecule itself, holding the RNA polymerase along the DNA molecule. And this alone, right, the alpha, the beta prime, and the omega subunits it's a functional RNA polymerase, just that. Notice I haven't talked about this sigma subunit yet, but we will. It will transcribe anywhere, but it needs to know where to begin. So if you can't just uh, it wouldn't be very efficient for a cell to just simply transcribe all the DNA all the time. There's regulation that happens, and that regulation happens in part because of this next subunit that we'll talk about, the sigma factor or the sigma subunit, and that's indicated here in this purple bluish subunit here. And notice that they show this -35 and -10. That refers to the promoter location. So the sigma factor, the sigma subunit, binds to that promoter region during initiation. It melts the H bonds, or in other words, it separates the DNA strands, and then it detaches. And this guides the RNA polymerase to promoter locations upstream of genes. So the question I put here in the slide, why bind at both places? Why -35 and -10? Well, if you have two locations, you can orient going the right direction. You can get this molecule facing the correct direction. If you only had it bind to one position, then the molecule wouldn't necessarily know to go left or right, to go upstream or downstream. But because it sits on the -35 and -10 position, that's what's thought to orient the molecule in the correct direction to move downstream from the promoter. So just to be clear on the definitions, the core enzyme are all of these subunits, alpha, beta prime, omega, and what's known as the holoenzyme or the whole enzyme, that would include the sigma factor. All right. Sigma factors are interesting because there's actually several different types of sigma factors. So it makes sense when you think about it that you would possibly want to turn on expression of a lot of different genes, say in response to some event happening. And for example, if there's a sudden thermal change in bacteria, they'll activate a bunch of genes at once to help deal with that. So heat shock proteins are nearly all switched on by sigma factor 32. So sigma factor 32 recognizes those consensus sequences. And others bind to genes involved in motility and chemotaxis or other types of genes. So there's different sigma factors that can bind to different types of consensus sequences. So we'll talk a little bit about now these three stages of transcription: initiation, elongation, and termination. And we've already really talked about initiation. So initiation is when the sigma factor binds to a promoter and tells the RNA polymerase where to go. And now you can then elongate the mRNA molecule. So just as a reminder, so a ribonucleotide is very, very similar to a DNA nucleotide, a deoxyribonucleotide. And of course, for RNA, you have uracil instead of thymine. But they are, in order to create a complete polymer, you have to have these raw materials, and one of these are nucleosides. And so this is a ribonucleoside triphosphate, showing the 3 phosphate groups, and the energy for polymerization of mRNA is obtained from those the inherent energy in those phosphate bonds. And so as this figure shows here down here, the RNA polymerase can move, unwind the DNA, use the energy that's contained in those phosphate bonds to string together now a series of nucleoside triphosphates to make one long RNA polymer. So now we'll talk about termination. How do you stop this from happening? There's two different ways, and this happens in prokaryotes when an RNA polymerase reaches what's called the terminator sequence. Depending on which sequence is encountered, the mechanism for termination is different. So the first way, this is conceptually the easiest one. So as the RNA polymerase moves along the DNA, and as that new mRNA molecule is produced, if a sequence is encountered that looks like this, that has a lot of complementary base pairing in that arrangement, then the RNA molecule itself, the mRNA, will take on this secondary structure and form, looking up in here, this hairpin loop. And especially, you'll see the typical termination sequence. This type of termination sequence contain a 40 base pair region of that's GC rich, has lots of G's, lots of C's. These tend to bind to each other, and it causes this hairpin loop to form. And when that happens, this causes the RNA polymerase to fall off. It can't move forward because this just physically kind of obstructs it. The second method is a little more complicated, also a little more, I think, interesting. I think it's easier to see in a video, and so I'll describe it here with some text and orally here, but I think it'll be interesting if you would watch this YouTube video that I've linked to, and I think that'll give you a probably clearer idea of what's going on with this. But the second method is called row-dependent termination, and the termination sequence here that's produced. So the mRNA is chugging along and it makes some mRNA sequence. It's a termination sequence. And it just happens to be a sequence that binds to a protein called the Rho factor. And the sequence itself, this type of termination sequence, is called a Rho utilization site. And a protein that's floating around in the cytoplasm will bind to that Rho utilization site in the mRNA molecule and kind of think of it as chasing down. The video, I think, will show it. Will do a good job showing this. But that Rho, then that protein, moves along the mRNA towards the mRNA polymerase, and it uses energy from ATP to move. And when it catches it, it does functionally the same thing as that hairpin loop does. It just sort of gets in the way, the RNA polymerase falls off. So again, this type of termination also is sequence dependent. But in this case, the sequence that's made into mRNA is recognized by this Rho protein. And the Rho then attaches to the mRNA and chases down the RNA polymerase and causes the whole thing to fall apart. So I'll leave this for you to do on your own if you want to do this as a practice question. So diagram a DNA, diagram the DNA molecule, include things like a promoter, 5' untranslated regions. This is a way to think about the structure of a gene. So if you want to do that, you can pause the presentation, look at the slide deck, and do that as practice. Another practice question, suppose that a consensus sequence in the promoter of a gene that encodes some enzyme, it's called enzyme A, was deleted. Which of the following effects would result? So you can pause the narration and think what you would answer. A, enzyme A would have a different amino acid sequence. B, mRNA for enzyme A would be abnormally short. C, enzyme A would be missing amino acids. D, mRNA for enzyme A would be transcribed but not translated. Or E, the amount of mRNA transcribed would be affected. So the best answer is E. The amount of mRNA transcribed would be affected. And the reason that's the case is if you delete the promoter, there's nothing for RNA polymerase to find. And so you're just simply not going to make very much of that mRNA because there's no way for RNA polymerase to sit down and begin transcription. Wouldn't have anything to do with changing the amino acid sequence or starting and stopping prematurely. It just probably wouldn't make a lot of mRNA. So there's some differences in transcription in prokaryotes and eukaryotes, and part of that relates to where the DNA itself is. In prokaryotes, there's no nucleus, of course, and so transcription happens in the cytosol. So you're seeing here DNA being made into mRNA, and then and actually what immediately happens is as soon as the mRNA is made, the ribosome can attach and starts making this amino acid polymer, polypeptide. In eukaryotes, it's a little bit different. So eukaryotes, of course, have a nucleus, and so the transcription occurs in the nucleus. And so DNA, of course, gets made into mRNA, but the mRNA here is referred to pre-mRNA because it needs some processing in order to be read at a ribosome first. And this processing occurs in the nucleus. We'll talk a little bit about what that is. So it's processed, and you'll notice that it becomes a little shorter. It also gains a cap on one side. That's an arrow. And a tail on the other side. So as it's processed, it becomes shorter, it gains a cap and a tail, and then it's exported out of the nucleus where a ribosome can bind to it and begin translation. So it's more complex. It requires more accessory proteins to get started. There's a video here showing some of that I think is worth watching. So I'd recommend that you click over and watch that before we keep going. So in prokaryotic transcription, there was a promoter sequence at -10, -35. Sigma factor recognized that, brought that RNA polymerase into association with the DNA molecule, and transcription started. Well, eukaryotic transcription is more complex, needs more accessory proteins to get the process started. And you might wonder why. And one thought as to why that is it allows for it to be a much more regulated process. So you could, for example, have a situation where in a eukaryote, it would only make mRNA if a bunch of different conditions were met. And if those conditions were communicated, through the production of other proteins. So for example, maybe you only want mRNA to get made at a certain point in an organism's life cycle, in a certain cell type, in a certain limb, or in a certain organ. And maybe even then only if nutrient conditions are right and the timing is right. And so more accessory proteins allow you to have more control of when an mRNA transcript gets produced. And some of those regulatory molecules are called transcription factors. And if you watched the video, you saw this. So eukaryotes require kind of this group of proteins to get together, and these are called general transcription factors. Sometimes they're abbreviated GTFs. And these have to all bind to the promoter before RNA polymerase can synthesize mRNA. Notice the mRNA polymerase 2. There's actually several types of RNA polymerases. RNA polymerase 1 synthesizes ribosomal RNA. RNA polymerase 2 synthesizes mRNA, and RNA polymerase 3 synthesizes a lot of the regulatory RNA. So there's in eukaryotes have this, not only do they require general transcription factors, there's also more types of polymerases. And so this kind of continues on with the theme of some of the things that we've talked about in DNA replication and now transcription. Eukaryotes follow a similar pattern as prokaryotes, they're just more complicated. So in eukaryotes, these general transcription factors bind to the DNA before the RNA polymerase 2 holoenzyme binds, and then others arrive and bind afterwards. They recognize a certain region upstream of the transcriptional start site, similar to the prokaryotic promoter. But in eukaryotes, this region is known as a TATA box. It has a lot of T's and A's upstream of the transcriptional start site, and it's outlined here in red. And it's thought that because it's a lot of T's and A's, it's easier to separate than, for example, a GC-rich region, because there's only 2 hydrogen bonds between the T and the A nucleotide in the strand. And proteins, one of the general transcription factors, transcription factor 2D, is one of those 6 general transcription factors. It contains a TATA binding protein, TBP. It binds to the TATA box upstream of the transcriptional start site about 30 base pairs. So again, similar to that prokaryotic promoter, about 30 base pairs away from where you're actually going to start transcription. This transcription factor TF2D binds and kind of anchors this whole complex, so a complex in the sense of, a group, this whole group of molecules that are going to together allow for that RNA polymerase to sit down. So the other than transcription factors, transcription factor 2A, 2B, and others, they all bind there to that promoter region or to each other, and then the transcription can start. So sometimes this is known as the pre-initiation complex. So these bound general transcription factors recruit or allow the attachment of, in other words, RNA to polymerase to core enzyme, and they position it to start so that it's facing the coding region of the gene. And I pointed out here that in eukaryotes, the RNA polymerase core holoenzyme has greater than 12 subunits versus the fewer that we saw, the 5 that we saw in prokaryotes. Lastly, and then this is a complex figure, and I pulled it from another. These are kind of cobbled together from several different textbooks, but this figure shows not only the promoter region, so these group of molecules here that are kind of all formed together, pre-initiation complex and the general transcription factors, but also a region far upstream, so far to the 5' side of what eventually will be the gene, which is encoded down here. On this 5' side, way up here, there's another sequence called an enhancer region. And this is showing what it's calling an activator protein. It's and really this can happen different ways, but just the idea is that you can have regulatory sites far away from the actual gene, but the only way that transcription will start is if a protein has recognized, for example, in this case, the enhancer region, and a co-activator has recognized this other regulatory site, and the general transcription factors have all bound to the Tata box region upstream of the gene. All of these things have to be in position in order for that RNA polymerase to kick off doing its job, and you get transcription then going that way. So again, kind of a complicated figure, but what it's trying to do, and I think it does a pretty good job if you sit and stare at it for a little bit, is it shows that a number of regulatory sites are necessary. Some of these are called enhancer sites, for example. This allows for, again, combinatorial control of gene expression. So this idea that many proteins have to be present in the right combination at the right time in order to express the gene to make mRNA. And sometimes whole other protein complexes are involved that are far away from the actual gene itself. So initiation in eukaryotes happens once the RNA polymerase is released from this pre-initiation complex, and then it can do its job. So this happens particularly in the video show this when you phosphorylate this carboxyl tail domain of the RNA polymerase molecule. It's inside one of the specific subunits, one of the proteins that form that RNA polymerase. So again, RNA polymerase is made up of many different protein subunits, and one of them has this sort of tail hanging off that can be phosphorylated. And when it's phosphorylated, it typically doesn't want to bind as well to this group of other proteins, and it can go off and begin to do its work. So elongation then happens. You make your mRNA strand, and as that happens, then some of the processing that I mentioned earlier begins. The molecule is shortened, caps and a tail are added. So we can talk about that here for the next few slides. So specifically, the pre-mRNA processing involves the addition on the 5' side of the mRNA molecule a 7-methylguanosine cap. It also involves the shortening of the molecule in a process called splicing. This is removing what are called introns, and I'll show a diagram that shows that in a minute. And then lastly, it involves adding a tail, and the tail is formed by a whole bunch of of A nucleotides being added. So this is called polyadenylation. So the cap, a 7-methylguanosine cap, it's unusual. It's connected by a 5' to 5' covalent bond in the mRNA molecule. It protects, one of its functions is it protects the mRNA from degradation. So mRNA molecules that do not have this cap are pretty quickly degraded in the cytosol. And this cap is also required for ribosomal recognition of the RNA. So you can't have translation begin without this cap present because the ribosome won't recognize it. And there's some enzymes that'll only associate with RNA polymerase II, and because of this capping also. So this is showing now in this figure the RNA polymerase with that phosphorylated subunit tail, and then capping enzymes that are present that are going to add the cap to the 5' side. Splicing is the removal of introns. So the mRNA is shortened, and it does that by removing these regions that are non-coding in the mRNA in the sense that they don't get translated. So an exon exits the nucleus. You can think of it that way. An intron does not. Exons exit the nucleus. So introns don't end up coding for anything. They're just present in the initial pre-mRNA. They might contain regulatory regions. On average, there are about 2,000 nucleotides in mammals, but other species types can vary. And with that splicing and removing introns, also you get joining the exons then together. So in this figure, presumably they're showing in red the exons in this mRNA molecule. And eventually these regions will be kind of chopped out. So I'm drawing X's to indicate these regions being chopped out, and you would be left only with what would be indicated in 3 red regions joined directly together. So those would be the exons that are all present in the shortened mRNA molecule. Exons typically are a lot shorter, especially in mammals. They're only about 200 nucleotides long. So if you think about it, the mRNA can be much, much, much longer when it's first transcribed than after it's processed. So one question you could have, why would it be advantageous for a genome to include introns? There's a number of reasons why that might be advantageous. One is that it allows for, like many things, increased diversity. So it allows for more crossing over to happen that would even happen within a gene because those genes are pretty long when they have the introns present. So crossing over could happen. It can lead to more diversity. You can also have regulation that happens with those introns. There's a lot of reasons why it's potentially advantageous for genomes to include introns. So besides the capping and then the shortening through splicing exons directly together, there's polyadenylation and adding a poly A tail. So at the end of most genes, and this is again in eukaryotes, you get what's called a polyadenylation signal, which is often AAU-AAA or AUU-AAA at the 3' end of a gene. So transcription still occurs, past that point, sometimes up to 1,000 base pairs. But what happens is that an enzyme cuts the mRNA a few nucleotides, sometimes up to 30, past that poly A consensus sequence. So that AAU, AAA, just a few tens of base pairs after that sequence, a whole bunch of adenine is added, 150 to 200 adenine, and that's what's known as polyadenylation. or poly A tail. And this tail has a number of functions. One of its main ones is helping the mRNA exit the nucleus. If you don't have that poly A tail, mRNAs don't tend to exit the nucleus very well. So we talked about RNA processing, a cap and splicing, and then adding a tail. There's another function of splicing besides removing introns and shortening the molecule is derived from what's called alternative splicing. And the next figure will show a little bit more specifically what that means. But the idea is that you might have 3 different exons that are encoded in a pre-mRNA molecule, but you could put them together in different ways. You could have all 3 of those exons exit the nucleus, or sometimes you could have just exon 1 and 2 exit the nucleus, or just 1 and 3. So it allows for, even within the same gene, different final mRNAs to be produced that could then produce different proteins. So in the human genome, for example, there's about 20,000 genes that are encoded. But if you looked at what's called the human proteome, if you sequenced all of the polypeptides that are present in cells, you'd find more than 100,000 proteins present. So how is that possible? It's possible because each gene can be alternatively spliced to include a different number, different combinations of the exons that are present. So the next page will show one example of a gene where there's known to be a lot of alternative splicing, and that's in a gene called tropomyosin. You might have seen this if you've taken an anatomy and physiology course, for example. So on the very top is kind of a lot of information in this figure, but if you look at this very top, the primary pre-mRNA transcript, it contains from left to right a whole bunch of exons. So 1A is one exon. These regions here in this light blue are introns. So 2A is an exon, 2B is another exon, 1B is another exon, 3 and 4 exons. But if you look and see how they represented this, in skeletal muscle or striated muscle, that's where we usually think of tropomyosin being expressed. That form of tropomyosin doesn't contain all the exons. It contains exon 1A, 2B, 3, 4, and 5. But for example, it doesn't contain exon 2A. It doesn't contain exon 1B. If you look farther down, the tropomyosin, one type of tropomyosin that is expressed in the brain, it doesn't contain any exons until 1B and then 3, 4, and 5 and a few others, and then none of these up here. So that means that the tropomyosin, it's called an isoform, that's expressed in the brain is different than the isoform expressed in striated muscle. They're both tropomyosin, they're both coded for in the tropomyosin gene, but the proteins do different things. They're differently shaped because, when they're translated, one is a lot bigger than the other one. And that because of that, they have different functions. So just to be clear, what's the difference between splicing and alternative splicing? They're very similar. Splicing usually just refers to the removal of introns to shorten the transcript, pre-mRNA transcript. Alternative splicing refers to this sort of mixing and matching of all the possible exons that are present in a gene to come up with different types of mRNA that can be used to code for proteins. There's some good videos on this on splicing. Splicing takes place, again, inside the nucleus and happens to pre-message RNA, pre-mRNA molecules. These 2 videos that I'm linking to here, there's sort of a general one and there's a pretty specific one with a lot of detail. the takeaway message from the videos, I really encourage you to watch it because it gives you kind of an idea of what's going on, is that this occurs at a structure called a spliceosome. It's about as big as a ribosome in the nucleus. And this is splicing machinery. It requires a lot of different proteins and RNA complexes, and it works by recognizing particular intron sequences. So sequences of messenger RNA, but in the intronic regions. And so the spliceosome recognizes those, cuts out the introns, and joins the exons together.